ever to make the world green and clean. No insects, no crop, no human. Insects ever dominate. No. Dominate our planet.
Okay, मुझे बोलने
बस
दीजिए।
कुशल
सर वाईके प्रसाद सर को बुके देने के लिए सुभाष सर को बुलाएंगे
और सुभाष सर को
हाँ।
देने के लिए सर को बुलाएंगे।
ओके
और आपको बुके चाहिए?
Sir is the host. Insect, the
You can
little
call,
wonders
Sachin.
that light the world.
Sachin.
सचिन सर को देने के लिए बुला लेंगे।
इधर से आना है ना,
फिर सर
हाँ।
इधर से आना
हाँ, इधर से आना, नो कोई दिक्कत नहीं।
फिर बुक के वापस कर देना सर नेक्स्ट
ए, नो
सेशन के लिए।
ऐसे नहीं है।
अर्चना दीदी
ए, नमस्ते।
सुभाष आई, सुभाष सर बोलेंगे ना, मैं बच्चों से बोल रही सुभाष सर को।
ठीक है।
आजा
सर मुझे अभी लास्ट मोमेंट ज्यादा
आयु अर्चना
little wonders.
वेलकम तुमने कर दिया ये सब कुछ,
ये तो नॉर्मल
है। उसके बाद तुमने वेलकम करेंगे, वेलकम में मुझे अभी बहुत ये हो रहा है ना।
ये सर का फॉलो करो।
आपको नहीं रहा इन्वोकेशन सब हो गया। सुभाष सर से बोल रहे हैं तो सुभाष सर फिर सुभाष सर आई एम वेलकमिंग द गेस्ट ऑफ ऑनर,
वेलकम द गैदरिंग एंड टुडे इज आवर चीफ गेस्ट डॉक्टर वाईजी प्रसाद। ऐसे मैं बोल दूंगी, फिर सर आएंगे अपना बोलेंगे। सर को मैं बोलूंगी, it gives me immense pleasure to invite Dr. Subhash Chandra sir to welcome the gathering and introduce today's chief guest, Dr. YG Prasad, Director, CICR Nagpur.
हाँ सही है, हो गया।
अब सर आएंगे। सर
सभी
बोलेंगे कुछ अपने गेस्ट के बारे से बताएंगे। गैदरिंग को
देंगे ना फर्स्ट, वो वेलकम में वहाँ ही बुके देते हैं। सुबह सर जब वेलकम करते हैं ना तब हम बुके देते हैं। बैठेंगे सर, हाँ तब मैं बच्चे को भेज दूंगी।
वो मैनेज कर लेना वहाँ से। हाँ
मैंने बच्चे को बुके रेडी करना।
सर आएंगे ना, एक बार बोल देना।
हाँ मुझे सुबह सर से बात।
ठीक है मैम, अभी मुझे फोकस करने दो।
वो है ना उसके टाइम में बुके लेके दे दिए।
नमस्कार। अभी कुछ नहीं। Insects. Thank you, sir. Insects, the little wonders that lights the world, ever to make the worlds green and green. No insect, no crops, no human. Respected dignitaries, ESI awardees, entomologists, and fraternity across the India. Respected dignitaries, ESI awardees, entomologists, and fraternity across the India, I, Dr. Archana Anokhe, scientist, Division of Entomology, welcomes you all on behalf of Entomological Society of India on this auspicious occasion of ESI Award Ceremony and first Entomological Society of India annual lecture. It's my pleasure to welcome the dignitaries to the dais. Today's our guest of honor, Dr. YG Prasad, Director, ICR, Central Institute of Cotton Research, Nagpur. I welcome you, sir, on the dais. Welcome, sir. I feel privileged to invite Dr. Subhash Chandra, sir, Director, NCIPM, to the dais. Welcome, sir. It gives me immense pleasure to invite Dr. S. Subramanian, Principal Scientist, Division of Entomology, IRI, New Delhi, to the dais. Welcome you all, sir. I request Dr. Subhash Chandra, sir, to welcome our guest of honor with the bouquet of flower. Welcome, sir. I request Dr. Subramaniam, sir, to welcome our Subhash Chandra, sir, with the bouquet of flower. I request Dr. Sachin Suresi, sir, to welcome Dr. Subramaniam, sir. Thank you, sir. Welcome you all on the today's occasion. I also welcome to our dignitaries and audience who has been connected through the online mode. Now, I would like to invite Aishwarya and team for the invocation song.
गणनायकाय गणदैवताय गणाध्यक्षाय धीमहि गुणशरीराय गुणमण्डिताय गुणेशानाय धीमहि गुणातीताय गुणाधीशाय गुणप्रविष्टाय धीमहि एकदन्ताय वक्रतुण्डाय गौरीतनयाय धीमहि गजेशानाय बालचन्द्राय श्रीगणेशाय धीमहि एकदन्ताय वक्रतुण्डाय गौरीतनयाय धीमहि गजेशानाय बालचन्द्राय श्रीगणेशाय धीमहि गानचतुराय गानप्राणाय गानान्तरात्मने गानोत्सुकाय गानमत्ताय गानोत्सुकमनसे गुरुपूजिताय गुरुदैवताय गुरुकुलस्थायिने गुरुविक्रमाय गुह्यप्रवराय गुरवे गुणगुरवे गुरुदैत्यकलक्षेत्रे गुरुधर्मसदाराध्याय गुरुपुत्रपरित्रात्रे गुरुपाखण्डखण्डकाय गीतसाराय गीततत्त्वाय गीतगोत्राय धीमहि गूढगुल्फाय गन्धमत्ताय गोजयप्रदाय धीमहि गुणातीताय गुणाधीशाय गुणप्रविष्टाय धीमहि एकदन्ताय वक्रतुण्डाय गौरीतनयाय धीमहि गजेशानाय बालचन्द्राय श्रीगणेशाय धीमहि एकदन्ताय वक्रतुण्डाय गौरीतनयाय धीमहि गजेशानाय बालचन्द्राय श्रीगणेशाय धीमहि
Thank you, Aishwarya and team, for the beautiful song. The Entomological Society of India was founded 1938 and it is one of the largest professional society in India, serving entomologists and researchers in related discipline. The society is publishing Indian Journal of Entomology since 9 decades. As per the recommendation of Executive Committee, the Entomological Society of India has introduced ESI Young Scientist Award in the 2020, and the society has introduced ESI Senior Scientist Award and ESI Best PhD Thesis Award from 2021 onwards. Accordingly, every year, 3 young entomologists, 2 senior entomologists, and best PhD thesis will be awarded to encourage, recognize, and promote the outstanding contribution of the entomologist in the country. With this, I would like to request Dr. Subhash Chandra sir to welcome the gathering and introduce today's guest of honor. Welcome, sir.
Thank you, Archana. Very good morning to every one of you this fine day. Our guest of honor, Dr. Baiji Prasad, Director, ICR, Central Institute of Cotton Research, Nagpur, and my old friend. We were together in IRI. There was 1 year difference. He was 1 year after me, and we spent good 5-6 years. So I am very, very happy to welcome you, sir. Very distinguished scientist. Dr. Subramaniam, Principal Scientist, Division of Entomology, and very distinguished scientist and convener of today's function. All the dignitaries, galaxy of entomologists were present here. All the members of previous executive, current executive, all the life members, scientists, faculty, students, ladies and gentlemen. It is my proud privilege to welcome you all today on the occasion of this ESI annual workshop 2022. which is marked by two very important events. Entomological Society of India Awards 2021. So their awardees, they will be making their presentations. And first, Entomological Society of India Annual Lecture 2022, which has been, for which Dr. Chandish Balal, she has been nominated, and I congratulate her for her nomination for this. As you know, our ESI has grown quite old. It is 84 years old now. So octogenarian, we can call. So very, very experienced. And it has very well fulfilled its mission of disseminating entomological knowledge, also provided platform for conducting meetings, discussions, seminars, and also have been publishing very important publication, Indian Journal of Entomology, Bulletin of Entomology, and also recently we have also initiated some other publications which are serving the community and therefore doing service for the country. So during all these years, very, very renowned personalities, they have been associated with our society, and we have been fortunate to have their, guidance and advice. So today also, I feel privileged in heartily welcoming a galaxy of eminent entomologists who have made our community pride, brought pride to our community while excelling in their professional careers and achieving, highest echelons of professional leadership as vice chancellors of state agriculture universities, central agriculture universities, and also deputy director general, directors of ICR institutes. So really, sir, you have done entomologist pride, and we are very, very happy to have all of you here. So therefore, I welcome Dr. Baiji Prasad, Director CICR, and guest of honor for the function, Dr. S.N. Puri, immediate past President, Entomological Society of India for 18 long years. Sir, you have done very, very, committed job to the society and for its betterment, and former Vice Chancellor, Central Agriculture University Imphal for 2 terms. Sir, with welcome you. I also welcome Dr. B.B. Patil, former Vice President and former Vice Chancellor, University of Agricultural Sciences, Raichur, sir. Welcome, sir. I also welcome Dr. Chandish Balal, former Vice President and former Director, NVIR, and today's distinguished speaker. And I also congratulate Madam, you are also done for you. Dr. B.B. Ramamurthy, President, Entomological Society of India, and former Chief Editor for 8 long years. Sir, you have done remarkable job for the betterment of the journal, for regularizing it. And so we thank you and welcome you, sir. Dr. K.S. Khokhar, I think he couldn't come, former Vice President and former VC, Chaudhry Charan Singh, Haryana Agriculture University, Hisa. We also welcome him. Dr. J.P. Singh, former General Secretary, Entomological Society of India, and retired principal scientist. Other members of former executive, Dr. M. Premjit Singh, former Vice President and former VC, CIU Imphal. He couldn't join. Actually, last week, I met him in Lucknow, so he has gone to Bangkok. So he has sent all his good wishes for this meeting. Dr. N.K. Krishnan Kumar, former Vice President, Entomological Society of India, and DDG Horticulture, ICR. He is also busy somewhere, so we also welcome him. Dr. T.P. Rajendran, former ADG. Sir, we welcome you. Dr. Jagdish Bentor, he was head plant protection and also PI for entomological at Indian Institute of Rice Research. We also welcome him online. Dr. K.L. Srivastava, retired principal scientist, entomology. Dr. T.M. Manjunath, so very distinguished entomologist. We also, Dr. Ram Singh, we also welcome you, sir. He's a very distinguished scientist from HAO. Dr. Sushil Kumar Saxena, Dr. S.N. Sushil, Dr. R.K. Anand, also welcome you, sir. I also welcome other members of former executive, Dr. H.K. Singh, Dr. Sachin Suroshe, who were former councillors, and Dr. Anam Meshram, our treasurer. So he has been transferred now from here. And all the members of newly elected executive. I also welcome them. Especially, I welcome the awardees and heartily congratulate them for splendid achievement. Achieving among senior entomologist awardee, Dr. Ankita Gupta, she is there online. Dr. Mukesh Kumar Dhillon, principal scientist, division entomology. Among young scientist awardee, Dr. Amalendu Ghosh, welcome him. Dr. Guru Prasanna Pandi, Dr. K. Silvaraj, Dr. P.R. Shashank. In fact, two of them are my students. Dr. Guru Prasanna Pandi and Dr. K. Silvaraj. They have been my students in PhD. And best PhD thesis awardee, Dr. V. Karyana. So, heartiest congratulations to all of you. I also welcome other project directors, heads, professors, students, and other attendees from various institutions who have joined online or offline. So, sir, I want to mention here that this institution of these various awards and this annual lecture, they are very good initiatives taken by the Entomological Society of India, and they will go a long way in encouraging our researchers and scientists to undertake innovative and competitive research. Because this time is of problems, so everyone is talking of innovations. Something new should be done in your research. Problem solving research. A lot of problems are there in the area of plant protection. So something with this, we should come out, our students, our researchers should come out something new ideas, problem solving research. So for that, I think these awards will prove very, very fruitful. And I can say one more thing, that entomological science is a very good, means a lot of applications in crop protection and in agriculture, but we can still value add to our research to adoption of various techniques and technologies, which are offered by molecular techniques, geospatial techniques, computational sciences. So we should all absorb them and add value to our entomological research. So I think that will go a long way in putting this entomology on higher pedestal among the agricultural sciences. So with these words, I finally welcome each and everyone who are present here and others who have joined online, whose name I might have missed. And I wish and hope that deliberations will be scientifically very, very fruitful and very good for especially for the new students. So I have also been assigned the job of introducing our distinguished speaker today, Dr. Baiji Prasad. And Dr. Baiji Prasad, he did his BSc Agriculture in 1981 from Baptala Agriculture College, and did MSc under the chairmanship of Dr. Y.S. Singh during 86 to 88 at IARI, as I was founded also PhD under Dr. Ramakrishnan, at time doing on genomic characterization of Spodoptera luteura nuclear polyhedrosis virus. And during his work, he worked on effective strains of BT, Nomuria relay, and Achia genata granulosis virus at Directorate of Oilseed Research. Hyderabad. And this technology was commercialized to 48 private firms. And the YSA protocols he developed for this insect, they were adopted by CIVRC. So very, very important research he did that time at Directorate of Oilseed Research. After that, he shifted to CRIDA as a senior scientist. And at CRIDA, he led multi-institutional consortium projects funded by World Bank, NATP, on weather-based pest forewarning in 6 crops, and NAIP project on development of decision support systems for IPM, and also developed and hosted model-based crop pest decision support system. He also developed the wireless sensor-based IoT application that time for pest and disease prediction in groundnut in collaboration with CDAC, Hyderabad. He was consultant for WWF India for farmer participatory technology development project on sustainable cotton initiative in 50 villages of Telangana for 6 years. And support provided for this project eventually led to the launch of Better Cotton Initiative in India. At CRIDA, he also coordinated 2 mega initiatives. Apart from his professional entomological research, coordinated countrywide efforts on preparation of district level agriculture contingency plans for 580 districts to meet aberrant monsoon weather situations as convener. Also coordinated the technology demonstration component of NICRA project, operated through 121 KVKs across the country before he became director of Atari, I think, yeah. And commensurate to this work, very, very deservingly, he is a recipient of Netherlands government NUFFIC international scholarship and trained in plant biosafety at ISC, Wageningen, and also attended training on pest modeling for outdoor crops at HRI Warwick, UK. He was also awarded Dr. Bap Reddy Memorial Award 2012 for IPM by Plant Protection Association of India. And he is a valuable partner in achieving the Golden Globe Award for Crop SAP project by Ministry of Information Technology, Government of India, which is also being implemented at NCIPM also. We are also partner in that. He is a member National Advisory Committee on Climate Smart Agriculture, study by TIFAC, DST, and chairman of PRSG on Annapurna Krishi Prasar Seva, which is an interactive ICT interface for scientists and farmers, piloted by the Digital India Corporation, Ministry of IT. Currently, he is a PRSG member for digital solutions for empowering farmers in Northeast. So what we are talking of now, digital sciences here, very early he has started this work. So you can see his foresight and, need of the day, what we needed. He served as director ICR Atari at Hyderabad, at CRIDA itself. I think it is was done since 2015, and coordinating 71 KVKs in the states of AP, Telangana, Tamil Nadu, Puducherry, and also initiated first on-farm trials through 19 KVKs on assessment of IPM modules for new invasive fall armyworm on maize during 2018, and also established 10 KVKs under his jurisdiction in 4 states under the chairmanship of Dr. S.N. Puri. He also visited abroad and presented his research contributions in international conferences in Netherlands, Malaysia, UK, France, Brazil, Indonesia, and Egypt. And he is a member of many, many professional societies and has to his credit 158 national and international publications. Currently providing leadership in cotton research and development as Director CICR Nagpur since 2020. So very important institute, and cotton is a very important crop. So sir, we, wish and hope that we'll get, under your leadership, able leadership, sustainable solutions to our pest problems, and our economy will, get enhanced. And we look for your advice and, leadership, and you will act as an inspiration for our young scientists and entomologists. So with all this, thank you very much giving me this opportunity.
Thank you, sir. With this, I would like to invite Dr. Timanna and team for playing ESI documentary. Invite Dr. Timanna and team for playing ESI documentary.
Before 17th century, Indian researchers were not getting proper recognition and serious attention for their discoveries and investigation in the field of entomology. Then, the first paper of entomology in India, that is, Koenig's article on termites, published in the research activities of Berlin Society of the Friends of Nature. It amazed the researchers of insect science, which elegantly matched the present understanding of termites and the British government appointed Lionel D. Knightsville as the post entomologist. Then
Holland, wherein he mentioned the existence of Institution of Civil Engineers, Chemical Society, British Science Association, and highlighted the advantage of having one entomology society for the whole of India.
he quoted, I do not know, however, to what extent the formation of an entomological society would meet the wishes and requirements of entomologists in India. Nor, I must confess, do I quite see what would be the function of such a society. My scheme for centralization of entomological work in India eventually. Such a central institute and service would fulfill generally to other workers. Even as it is, our periodical meetings provide ample of opportunity for discussion of any problem, and it is difficult to see how more frequent or better attended meetings could be arranged in such a vast country as it is. Nor is there any lack at present of facilities for publication.
This tradition, mooted by Fletcher, hibernated for 18 long years. But the aspiration thought of T.V. Fletcher was again re-emerged during the 24th Indian Science Congress held in Hyderabad in 1937. The formation of Society for Indian Entomologists was done by forming an ad hoc committee consisting of Muhammad Afzal Hussain, Hem Singh Pursi, T.V. Ramakrishna Iyer, Y. Ramachandra Rao, N.C. Chatterjee, Devraj Mehta, and Durga Das Mukherjee to formulate rules and regulations for the society. On 7 January 1938, at the Indian Science Congressation in Calcutta, C.V. Ramakrishna Iyer moved a resolution that the Society for Indian Entomology, named as the Entomological Society of India, and the draft rules developed by the ad hoc committee was accepted unanimously. Entomological Society of India was formally launched by Afzal Hussain, an eminent locust entomologist in India, and the principal of the Punjab Agricultural College Mathayar.
In the context of the just inaugurated Entomological Society of India, the founder president, Afzal Hussain, says, it is most necessary for the future development of our science that a powerful independent body of scientific opinion be created to foster the growth of entomology in our country. An entomological society with branches all over the country is greatly needed. And at the time of inauguration in 1938, the Entomological Society of India enrolled 42 full members and 1 associate member. In 1938, ad hoc committee determined the society's objectives.
Most of us may not be knowing the curious story behind the selection of live insect as the society logo. In 1835, a person called William Wilson Saunders exhibited males and females of live William volumes collected from India at a meeting of the Entomological Society of London. The curious morphology and its endemicity to the Indian subcontinent attracted the Entomological Society of India office bearers in 1938 to select the insect. The branches of Entomological Society of India were established to reach out and coordinate entomological researchers from different parts of the country: New Delhi, Sindh, Punjab, and Coimbatore. As the year passes, branches were changed to chapters at Coimbatore, Madurai, Umiam, Akola, Parvani, and Latur.
During those days, the Honorable President of India were praised society role in safeguarding food security and public health. The establishment of Entomological Society was over, but the next challenge was publication. The founder president, Muhammad Afzal Hussain, emphasized the necessity of publication for entomology research. The editorial board comprising H.S. Kruti, C.V. Ramakrishna Iyer, N.C. Chatterjee, D.R. Mehta, Khan A. Rahman, Krishna Behari Lal was constituted. Kruti was the chief editor. The first issue of Indian Journal of Entomology rolled out of the press in June 1939. Entomological Society of India organizes various international and national conferences, symposia, workshops, meetings to promote the entomological research of the India to the world. There were renowned entomologists who served as the president of Entomological Society of India. The Entomological Society of India has seen phenomenal growth in recent years. Under the stewardship of Dr. V.V. Ramamurthy and patronship of Dr. S.N. Puri as the president of Entomological Society of India from 2004. Currently, annual membership of Entomological Society of India reached more than 2,500 with 626 life fellows. Entomological Society of India also publishes other publications like Memoirs of Entomology, Bionotes, Bulletins of Entomology, and Indian Entomologist magazine.
However, the changing time required adoption and improvement in communicating insect science to the people. Entomological Society of India has been modernized to reach researchers by moving to open access from 2020, which enables easy accessibility. To encourage and promote entomological research in India, Entomological Society of India has launched Entomological Society Awards for scientists and best PhD thesis started from 2020 by recognizing their outstanding work in the field of entomology. Entomological Society of India
Thank you, Dr. Thimanna. Hemant and team for wonderful compilation. It is very relevant to this context that it is a man who want to create a history, should know the history. So thank you for tracing the entomology history. With this, I feel privileged to invite dignitaries of the dais for releasing the ESI souvenir on these occasions. Thank you, sir. Thank you very much. Now, I would like to invite today's guest of honor, Dr. YG Prasad, sir, for introductory remarks and preside over the sessions.
Yeah. All the dignitaries here, all the online participants, good morning to all of you. Dr. S.N. Puri, former president of the society, Dr. V.V. Ramurthy, current president, Dr. B.V. Patil, former vice president and former vice chancellor, Dr. Subhash Chandra, director NCIPM, Dr. Subramanian on the dais, Dr. Chandish Ballal, former director NBIR, Dr. S.N. Sushil, who has joined online, Dr. R.K. Anand sir, Dr. Renu Srivastava, Dr. Manjunath sir, who has joined online, Dr. T.P. Rajendran, Dr. J.P. Singh, Dr. K.L. Srivastava, who has joined from Malaysia online. I am seeing him after a long time. Dr. J.S. Bentur, Dr. Ram Singh sir, all the executive members, former and current executive members of the society, all awardees, all professors, teachers, students of the division, and all entomologists and members of ESI who are participating in this physically as well as online. Good morning to everyone and a warm welcome to you for this ESI Awards and annual workshop. It is a great privilege for me to be here after so many years, a student of IRI, to be amongst you, standing before you and then participating as a guest of honor in this. Thank you for the society for inviting me. It's a great privilege and honor, and I'm very happy to be here amongst you and take part in this award ceremony. I have not much to say at this point of time. I just would like to invite the presenters later on, after the award ceremony. Maybe I'll have some scope for, as per the schedule, I'll follow. So should I invite now? We have about, we'll have the presentations now. So we have 2 plus 4 plus 1, 7 presentations. So first presentation, Dr. Ankita Gupta, I think she has joined online. Are
Yes,
you
sir.
ready, Dr. Ankita?
Yes, sir. Yes, sir. Yes, sir.
So she is ready, and then she is the Senior Entomologist Awardee. I now invite Dr. Ankita Gupta to be followed by Dr. Mukesh. Then we'll have the Young Scientist Awardees, Young Entomologist Awardees, Dr. Amalendu Ghosh, Dr. Guru Prasanna, Dr. K. Selvaraj, who is here, Dr. Shashank also here, and also the PhD thesis, best PhD thesis awardee, Dr. Karyana. All these presentations, we'll have the pleasure of listening to them, and thereafter, I think we'll have the award ceremony. Thank you very much. I invite Dr. Ankita to make a presentation.
Yeah, thank you, sir. Thank you, sir. So shall I share my screen?
Yes, ma'am.
Please let me know if the screen is fully visible.
Yes, ma'am, it is visible.
Yeah, a very good morning. Warm greetings to all the audience, respected audience there at IRA, as well as the online audience. I remain humbled and grateful to the Entomological Society of India to consider me wise enough for this award. So with this, I would like to first start my presentation with an apology that because of my personal commitment to attend cancer patient at my home, I could not physically come. However, I'll try my best to communicate my whatever work I have done. So I would be sharing in brief the journey with the parasitic wasps and the light taxa, which I have been working. Basically, I work on taxonomy of parasitoids. And initially, when I started, I joined as a permanent employee at PDBC. I started working on braconids, and I took microgastrinae as one of the subfamily to work. And the first and foremost thing which I did was to estimate the species diversity as to how much I have to, or what is remain undocumented should be known. So I took the model concept from the Nearctic region and the Neotropical region, which says that the normal Lepidoptera to the microgastrine or the parasitoid ratio of a well diversity documented region should be somewhere in and around in between 7 to 20 with an average of 12. So with this, if we calculate, we come to know that just when I made the checklist, only 1/4 of the species which is supposed to be known was known. So I moved ahead and I started my surveys, and I continued surveying systematically on a majority of the country areas I have covered. And during this process, I have described around 65 new species. Some of them are non-Indian species also. First report of 227 new host records, and revisionary studies of 4 taxa have done, one family, which is Vespidae, one subfamily Microgastrinae, and 2 species group of Braconidae from the Oriental region. Global review I have done for 2 taxa, for 2 genera, and species nomenclature changes I have given with 10 synonymies, 3 new combinations, and 1 species status revised. Indian checklist we have made for Vespidae. There was no Indian checklist for Braconidae and Vespidae, so updated Indian checklist has been made. I have added some 1,200 authentically identified species to the collections with around 20K identified specimens. Many more specimens are there with me in thousands, which I'm yet to identify. I have been continuously providing identification services and have published 126 peer-reviewed research papers. So my mode of collection has been passive as well as active. So I have used different traps, Malais traps and yellow pen traps, and as well as sweep nets. But however, I have been focusing more on host rearing, which has been a major mainstay in my group. So for this, I have initiated a transcontinental approach for studying Braconidae, in which we are a team of collaborators across the globe on different taxa, and we have started our work. Just to start with, the first work which I did for the phylogeny of one particular, I wanted to do the integrated taxonomy of a highly speciose group, Glyptopentalis, in which I had brought the first concept of clade limited host utilization pattern. And this led to the discovery of 8 species groups with 26 provisional new species, which I am in the process of describing. This was done with team NBIR with Dr. Venkatesan and group. And after that, side by side, I have been studying fauna from different parts of the globe. This was Afrotropical fauna, which was from the Reunion Island, which has one of the most active volcanoes on the earth, and this island is a hotspot for many of the naturalists to work on. So we have worked on the fauna of this place and given a catalog and 18 new taxa and a key to species. Then I have collaborated with Chinese workers, and we recently have done the revision for 2 groups. A majority of the species are Chinese and Indian, at a group of Apentalis with synonymies and almost like 97 species we have treated. For this particular work, I have examined 17 species type at Natural History Museum. I have done the redescription of 11 species. And now we can say which species are present in China as well as India together. And this is the first ever comprehensive study which has been done for any of the Apentellus group from Oriental region. The second one is Altor group of Dolichogenidia, in which we have treated 67 species. I have examined all the types at NHM, and we have redescribed few species and I have given you host records. Then along with other workers, we have worked on the review of Cotacea. Here, one particular subspecies, or we cannot say it's a subspecies, but there was a conflict in the major character. So that character, that is a mediotergite which tapers at the apex. I'll not go more into the taxonomic details. So that has been present only in Africa as well as India. So we have examined some 269 species, and we have done the parasitoid host wherever it was available, the analysis. And based on this, we have come to the conclusion that the molecular data of all the species, including the cryptic species which we were talking about, they support the monophyly of the genus Cotacea. Then with the international students, which I have got from Iran, we have worked on the palearctic fauna and 6 new species with review of 3 genera I have done from palearctic region. So again, with Team NBIR, Dr. Venkateshan, we have worked on the host range, which is redefining boundaries for Snellius and Microplatus, which are the 2 genera which now possibly with international collaboration, we are in the process of synonymizing them. Secondly, under the CABI project, we have dealt with the hyperparasitoids of the biocontrol agents of the invasive weeds Hedicium and Rubus, and I have described a new species from the Himalayan region. So then majority of my discoveries have been from the butterflies, where the Hesperids have been the most predominant host, and Braconids the most predominant parasitoids. And as you can see in Hesperids, 27 host species has yielded 31 species. Many of them are new in this. And this is one of the glimpse I'm giving for. I'm not going in too much into details because of time constraint. Then similarly for nymphalids, 7 host species has yielded me 6 species of wasps. Like canids, I have reared up to 16 species, which has given me 9 species of wasp as well as tachinids. Same is the case for pyrids. So now I can very confidently say that which particular genus has which kind of host range, and what is the exclusive host for some particular genus. That also we have published it. So with this, an exclusive solitary and gregarious species and the cocoon pattern formation specific to a species also has been published. So this is the category which I have demarked as reared. And so in this altogether, in this work, comprehensive work, I have gone into 200 morpho species, which I read from 75 host species belonging to 22 families. During all this process, we have got many new family host records, global records, which for the first time host association, including the rare family of butterfly Rhyodinidae, which yielded us parasitoid for the first time. So likewise, for many group of pests, we have documented all the parasitoids, egg, larval, and pupal, as well as for many of the arthropods. As you can see, some of them has yielded us new species, be it a hemipteran or a coleopteran. Many of them are new global records also. So some of the very interesting case of ectoparasitism and endoparasitism, new species clearly demarked as ectoparasitoids and endoparasitoids have been published during this process. And this particular work was done along with Dr. Selvaraj, who has given me the specimens of whitefly from one region. And we reported for the first time a family Zinesti, a rare family from the Indian mainland. And for the first time globally, any parasitoid of Zinesti was reported from the world. So we reported this. Then likewise for all the predators also, even from the nest, I have documented majority. Now I'm going to come up with a publication with all the parasitoids of the predators from their native nest there, where they're housing. And we have documented many of the parasitoids from
Dr.
the...
Ankita.
Yes, sir. Yes,
one
sir.
More, one more minute here.
Sir.
Okay, one more minute. Okay.
Sure, sir. Sure, sir. So I'll just skip this particular part. I'll go to this cassava mealybug. In this recently one work, what we have done, the team NBIR as well as with Dr. Shashank and all, we have taken the help in which we have read multiple predators which are present, and we have separated the predators, and we have gone to the parasitoids even up to the 4th trophic level and documented up to 45 species of arthropods which are present in CMB niche. Now at present, we are working on the parallel gill dynamics and the interspecific competition between the predators in the CMB niche. So during this, I have reported many of the invasive pests, their parasitoid complex. For the first time, I have initiated larval taxonomy after 108 years of its, and I have given new characters. And for this, the rearing was done up to 235 days for Excalifus dicax. And the conclusion of this is the species which is present in India is dicax, which had a confusion with the species festivus, which is present in Africa, Middle East, and this thing. So I just come to the end of my presentation. During this, I have been in, I attended a modern taxonomy program, which has given me a lot of insight. And for this, I got the EDIT grant and DST travel grant. I have been to a naturalist museum where I have examined many specimens. 9 databases have been prepared, which are hosted in NBIR websites. 3 technologies we have commercialized. This is under the guidance of Dr. Subharan. We together as a team for 3 species for Musca domestica management in poultry we have done. And this is the expert system what I have developed for the, now at present, we are getting many international students in the museum for training under parasitoids. So this is the last. I owe this award very much. I'm thankful to the Entomological Society of India, ICR, most importantly, Network Project on Insect Biosystematics, who has groomed me to whom I am today, Dr. Ramamurthy sir, who has ever been supportive. There was an incident when I wanted a camera and I just went, and sir, within a span of 5 days, the money from NPIB project has come and I could purchase the camera. Dr. Balal has been very instrumental. I owe a lot to her because she was instrumental in my NHM trip. Otherwise, it would have been not possible. Dr. Purni to share all the lab facilities with me, and definitely I have learned everything of taxonomy, what I know today, through her publications and the way she approaches her work. All the contributors and of the international contributors and the sponsors, SERB, INSA, CABI, CIRAD France, NHM, Iran and Nepal Ministry, and International Society of Hymenopterists, who have been supporting us. The only thing what I have gained, I feel as of now till in the journey, is that one genus was named on me by the CNC group, which I consider it as my privilege and a great privilege, and one species from University of Illinois. With this, I would like to end my presentation. Thank you, one and all.
Thank you, Dr. Ankita. Very nice presentation.
Thank you.
And congratulations to you
Thank
on
you,
being awarded this prestigious ESI award for senior entomologist.
Thank you.
Once
Sir.
again, congratulations and thank you. Be online.
Thank you, sir.
We'll have the next presentation by Dr. Mukesh Kumar Dillon. Dr. Dillon, please, 10 minutes time.
15 minutes as allotted, but I'll try to finish in 10 minutes.
Yeah. We can have some discussion because you have difficulty.
Yeah. Good morning, family entomology, including the senior veterans and dear friends. First of all, I would like to thank Entomological Society of India for recognizing the research contributions through this Senior Entomologist Award. Straightway, I'll be going for the presentations to save time. Can I have the control on the presentation? This front? No, it's not moving. No. Huh? Okay. Okay. Next one. Okay, my presentation will be in 4 different components. The first component will be on, yes, next. first component will be on to understanding the CMS systems and how these CMS systems can be used for insect resistance. And here we are, I will be talking about sorghum fruit fly as a case study. In the second one, insect diapause. And in that, the Chilopatrus will be one of the case study. And in all these things, because these were some of the important components which we need to understand to have a better strategies for the pest management. And in the third, how these BT crops in the pest management and what are their non-target effects on the non-target organisms, and also host plant resistance, insect plant interactions and other things next. Going for the first one, next. Going for the first one, CMS systems and their role in host plant resistance as a case study. So this study was conducted, and there are several such publications have come out of it. And the questions we asked about what are the mechanisms, what are the inheritance of resistance, and whether these CMS systems have any role or in the resistance or susceptibility of this pest. And next. So from this study, we could conclude that the cytoplasmic male sterility systems in sorghum influences the expression of resistance to fruit fly, and the fruit fly resistance traits are governed by additive type of gene action. And the hybrids of these CMS-based systems were genotypically and phenotypically closer to cytoplasmic male sterile lines than the restorer lines. And the another conclusion from these studies was the A4M Maldandy cytoplasm is comparatively resistant as compared to the other cytoplasmic systems, which can be used in sorghum hybrid programs. And at the last, resistance should, if we want to have the insect resistant hybrids in those, we should have resistance in both the parents. If not in both the parents, at least A4M cytoplasm, Maldandy cytoplasm can be transferred in the insect resistant, fruit fly resistant background, so that we can have better resistance as compared to the other. Next. And the other component, next. In the insect diapause. So this study also we have conducted over a period of time, and it has been published in several highly recognized journals. So from this study, we could understand what are the genetics of this diapause, whether there is any races, or you call it a different biotypes on this insect. And likewise, what are the effects on its physiology and behavior, etc. Next. So from these studies, we can conclude that diapause is although an adaptive mechanism to survive unfavorable conditions, but there is certainly a population sacrifice during hibernation and estivation. Even these are the studies from the laboratory, but we expect these under field conditions as well. When we are talking about the modeling, this Ekimoto and Takai, these are linear models, and lactin and lactin 1 and 2, these are non-linear models. These are the important models which temperature dependent development and estimating the thresholds of these diaposing Chilopatrus populations can be determined, and based on these prediction models could be useful for predicting the occurrence, seasonal emergence, or number of generations in population dynamics of this insect. Diapause has been found to have deleterious effects on this post-diapause development, reproductive physiology, and population growth. Also, from these studies, we could see that there is changes in the biochemical profiles of these diapause populations, and also about the inheritance of resistance. From that, we could understand that degree of dominance of this diapause. We could see that development and morphometrics of this Chilopatrus are governed by over-dominance gene effects. Mainly depends on the parental diapause history, and also suggests to explore appropriate genetic means for management of these populations under different agroecological regions. And another important finding from these studies was that we could see from that across the India, we have four different biotypes in Chilopetalus, and namely Hyderabad, Hisar, Parbhani, and Coimbatore. And for that, at least the test genotypes of either sorghum or maize, whatever the pest is. So all these four locations are important to test these against for these locations so that we can have the better resistance, stable resistance. So from temporal distribution of mating systems among adults of diapause, non-diapause, and or ecologically diverse populations and their behavioral and physiological consequences under a given demographic framework could be rewarding. And from this, we suggest to devise appropriating mating disruption techniques so that we can stress this population under field conditions. Next. Under this component, BT crops pest management and environmental safety to non-targeted organisms. From these studies, several highly rated peer-reviewed journals we have published. And from the main conclusions of these studies are next. Next, yeah. There are no direct effects of BT on the parasitoids or predators. Whatever indirect effects we see in these one are either due to early mortality or poor quality of the host or prey. The generalist predators are less exposed to the transgene products, as it is likely that not all preys are BT contaminated under field conditions because they have alternates. The transgenic plants have shown no adverse effects on the abundance of non-target insect pests, generalist predators, and parasitoids under field conditions. And if there are any, these are much lower than that of the insecticides. And the non-target beneficial arthropods, again, significantly lower in insecticide sprayed than that on the unsprayed conditions. So we can see how much loss to this biodiversity and natural enemies causes these insecticides. And in the last, the BT cotton as a case study, from that we could see that it reduces the bollworm damage, increase seed yield, and also these are safer to natural enemies as compared to that of insecticides. From all these studies, we could conclude this. Next. Insect plant interactions. This is a continuous process, and in that probably several good publications have come over the years. And from these studies, at least we can see that although we have deciphered several biochemical defense mechanisms against various insect pests, but understanding on biochemical defense and their metabolic pathways, the associated enzymes and gene regulation systems, biochemistry of host plant as well as that of the associated insect, and the metabolic
It's not audible now. Please put on the mic. Audibility issue, please.
Sir, I think WTC that there is a net connectivity problem is there, so that's why they have left from the meeting. They will again join, sir.
There is break in voice, audibility 0.
Morning, Rajendran sir. They are resuming it, sir. They are working on it.
Organizers, please. Nothing is audible. We lose track of presentation. Please do something. we online participants are not able to listen.
I invite the dignitaries on the dais. Okay. To the online audience, we are really sorry that there is a network issue in our system. So there were some interruption. Now we will start with the Young Entomologist Awardee. I'll give a stage to the sir.
Yeah, I invite Amalendu Ghosh, the Young Entomologist Awardee, to make his presentation. Can do it for 10 minutes and
Yes, sure, sir.
maximum 12 15 minutes.
Thank you, sir. So good morning. I'm Amalendu Ghosh from IRI New Delhi. And before I start, I would like to express my sincere gratitude to the Entomological Society of India, and I congratulate my fellow awardee. I basically work on, oh, shall I wait or?
Go ahead. Go ahead.
So I work on two major vector, thrips and whitefly, with the objective to understand the thrips virus and whitefly begum virus relationship, aiming to interrupt the virus transmission and restrict the spread of the thrips whitefly population. So I shall talk on the virus transmission by thrips, followed by begum virus transmission by whitefly, and then spray on application of dsRNA as a novel management option. In thrips virus relationship, the present understanding is largely based on the Frankinella occidentalis and the tomato spotted wheel virus TSWV. Little is known about the other virus and thrips system or complex. So we reported that the thrips palmi, that is our model organism, thrips palmi adult can only transmit the virus and the adult cannot become viviparous if virus acquisition is not taking place during the first instar larval stage. Although these tosmoviruses like GBNB and WNB have negative effect on the life cycle of the thrips spermie, the viruses are efficiently transmitted by thrips. They modify the thrips life cycle in such a manner so that the virus is efficiently transmitted. Now, in case of the dissemination of the tosmovirus within the thrips system, for successful transmission, this is the schematic diagram of the thrips alimentary canal and salivary glands. For successful transmission, the virus infection should reach the primary salivary gland. And in case of thrips, the primary salivary gland are directly connected to the foregut or anterior portion of the foregut with a pair of ligament and tubular salivary gland. So the earlier concept tells that virus from the midgut directly travels to the PSG through the tubular salivary gland. An adult cannot acquire the virus to become viviparous because TSG disrupt during the adult stage. So that is the current concept. So we tried to find out whether this is conserved in thrips spermie or different. So you can see this is the first one is the larval picture and second one is the adult picture, and this is the TSG and it is intact across the life stages of the thrips. So the earlier concept of disruption of the tubular salivary gland in the adult stage is not correct. So now question is then what is the primary channel of the virus dissemination from the midgut to the primary salivary gland? So we found the initial infection occurs in the epithelial cell of the anterior midgut, and from there it travels to the primary salivary gland, primarily through the connecting ligaments, not via the tubular salivary gland as described in the earlier reports. Next question we answered, that is the why cannot adult become viviparous if acquisition taking place in the adult stage? Basically, in the adults, the infection retained in the visceral circular muscle and the longitudinal muscles of the anterior midgut, and there is no infection in the epithelial cell during the adult stage. In fact, the epithelial cell in the thrips adult do not allow any fresh infection of the virus. So this is the susceptibility of the epithelial cell that actually determines the infection of the midgut only during the larval stage, but not in the adult stage. So this is the first evidence of tosmovirus dissemination path in the thrips spermie, and that also clarifies the age-old concept of life stage specific tosmovirus transmission by thrips. We also established one primary cell culture of the thrips spermie and demonstrated the localization of tosmovirus nucleocapsid protein in the nuclei of the thrips spermie cells in vitro. This cell culture, this primary cell culture serves as a platform for study the thrips tosmovirus relationship in vitro. In a computational analysis, we predicted that the invasion of tosmovirus in the cellular system of the thrips involved the clathrin-mediated endocytosis, and two proteins, the syntaxin 6 and UHRF binding protein 1, play important role in the viral fusion in early endosome of the thrips cell. So that was supported by the transcriptomic study, and out of these differentially expressed genes of the thrips in response to the GBNB infection, we targeted two genes. One is UHRA binding protein 1 and the PFAS of the thrips to silence using modified antisense oligos and see their effect. So that downregulates the target gene expression by 2 to 3-fold, and that also induce around 90% mortality in the thrips. And with exposure, the mortality reaches around 100%. And interestingly, the ASO treatment, antisense oligo treatment, create morphological deformities in the treated thrips spermie. The body become flaccid and abdomen flattened in case of UHRF1 binding protein 1 silencing. And when we silence the PFAS of the thrips spermie, the appendages get twisted, body become brittle and little bit squeezed. And most importantly, the silencing of UHRF binding protein 1 and PFAS reduce the virus copy number in individual thrips spermie by 200 to 300-fold. So this could be the potential genetic target for reducing the crop damage by the thrips, as well as it will also restrict the virus transmission. Now, coming to the Begum virus transmission by the whitefly, Begum virus are transmitted by whitefly in a persistent circulative manner. That means the virus does not replicate within the vector and there is no transoviral transmission in general. For the first time, in exception to that, for the first time, we reported the eggs and the next generation adult of the Bemisia tabaci contains the viral DNA of Dolichos yellow mosaic virus. Not only that, the next generation adult can also transmit the infection to the healthy plant and produce strong symptoms. The CY3 probe hybridization also prove the evidence of invasion of the virus in the reproductive organ of the whitefly and also in the eggs. So this is the first evidence of transoviral transmission of a bipartite Begum virus in whitefly. And also this finding has epidemiological evidence because in absence of the host plant, Bemisia tabaci itself can serve as an alternate host and bridge the gap between the cropping season and immediately available to infect the plants when they are germinated in the field. In a different study, we also reported the dynamics of two different Begum virus in and out of the whitefly during feeding. Although these viruses do not replicate within the vector, but their concentration increased due to the accumulation with time. We also tried to knock down the B. tabaci key genes by RNA and thereby restricting the transmission of the chili leaf curl virus. So based on the transcriptomic study, we targeted two genes of the whitefly HSP70 and the fasciclin-2, and the oral delivery of dsRNA downregulate the gene regulation by 8 to 12-fold, and that also induced a mortality around 70 to 80%. Interestingly, with the gradual increase of the HSP70 dsRNA in Bemisia tabaci, the viral concentration in individual whitefly started reducing. And at a highest concentration, there was no virus copies detected within whitefly. In contrast, when we silenced the fasciclin-2, the virus copies increased in the whitefly, maybe due to the negative regulatory role of fasciclin-2 in Begum virus transmission. In line with that, in case of plant-to-plant transmission, you can see the picture with the increased concentration of HSP70 dsRNA, the transmission gradually reduced, and at the highest concentration, there was no transmission under laboratory condition. So we thought whether this can be used like an insecticide and sprayed on the plant. So for that, we conducted one semi-field condition, and the HSP70 dsRNA was sprayed like an insecticide with a 10-time higher dose than the control condition. And that reduced the expression of the target gene by 4.6-fold. The virus copies was reduced by 10 to the power 9-fold, and there is about 75% reduction in the virus transmission from plant to plant. Although some plants were PCR positive, but they did not produce any symptom up to 50 days. That is very crucial phase of the crop growth because symptom appearance after that period does not have much significance in the yield reduction. The spray-on application of the HSP70 dsRNA also provide good control of the infestation of the whitefly up to 8 days, and the second spray on the 10 days further increase the window up to 18 to 20 days. So this spray-on application of dsRNA targeting the Bemisia tabaci HSP70 provides significant mortality of the whitefly and also restrict the spread of the Begum viruses. And this could be a potential eco-friendly alternative to the hazardous pesticide and can reduce the pesticide use. This is also alternative to the virus-resistant transgenic plant development program. So with this, I just finished my talk. I acknowledge the support I received from my institute, IRI, organization ICR, my funding agency, my collaborators, laboratory students, SRF, and laboratory staff. Thank you.
Dr. Amalendu, very nice presentation.
Thank you.
From basics to applied. I think end to end, your research has progressed in that fashion, and great compliments to you because as a young scientist, you are able to think about an application for a basic study. And that too solving a very important, intricate disease problems transmitted by insects like leaf curl and all virus problems. I hope you will work on CLCU leaf curl virus of cotton as well. Because the same Bemisia tabaci Asia biotype 2.1 is also the vector for leaf curl virus,
Yes.
which is actually damaging a lot. This season, expression was 30% of the plant incidence in North India, especially in Punjab. Great damage it is causing.
Yes.
I hope you will, or your group or some other group is already working on this.
Basically, sir, what we are doing, that is based on the that is a model organism, chili leaf curl virus and Bemisia ACR21. So that whatever findings we have, that can be extend to the other pathosystem also with little bit modification and optimization.
So if any urgent burning question is there, we can take one. Otherwise, we'll go for next presentation. Thank you, Dr. Amalendu.
Thank you, sir.
Dr. Guru Prasanna Pandi. Guru Prasanna Pandi. Young entomologist awardee. Presentation for 12 minutes or so.
Yes, sir. Thank you, sir. So, honorable dignitaries and most respected teachers, senior colleagues, friends, and my dear students, a warm welcome to everyone. First of all, I would like to thank the ESI for giving me this prestigious award and also giving me the opportunity to share my significant achievements in the field of rice insect pest management, some of the strategies we have developed, and I have learned some field learning. This presentation, I'm going to present in the 5 different subtopics, starting from post-plant resistance, then molecular entomology, climate change aspects, pesticide, and lastly, IPM. So when I started my career as a PhD student from IRI, then I started my scientific career from NRI. I have been assigned to work on screening of germplasm against brown blonde hopper, one of the notorious pests in rice. And following the standard protocol developed by IRRI and a little bit be modified for our conditions, we have screened different germplasm available at our gene bank as well as from the NPBGR gene bank and some of the material from ICRE. Till now, in this line of work can access in our website. It is readily available. And from there, you can see from the year for the particular germplasm screen year 2004 or 1960, that germplasm, what is the status now that we can see. Further, after identification of the resistance in the laboratory condition, we have studied the mechanism of resistance by these 4 characters in ontogenesis as well as ontobiasis, and again with the genotypic parameters. For this genotyping, I have selected 93 gene-linked markers spanning the rice chromosome starting from 1 to 12, covering 34 PPH resistant genes. Here I am presenting only a single case study. Here we took Odisha farmers variety, that is a panel of 600 variety. Upon this 600 variety screened for consecutively 3 years, we have selected 112 variety for genotyping analysis. And we found that through the genotypic analysis, it's found structure 3, K3, that our other analysis, like cluster analysis, also supported this study. I am pressing this issue here. The unique observation we observed here, 2 germplasm we identified as resistant source also present in the same cluster as of PTP33 and Salkati, a known BPS resistant sources. Further, after the phenotypic and genotypic parameter, we have put it in the general linear model and multilinear model to identify the resistant gene which are associated with BPH. Among the different genes has been reported till now, these 4 genes I have listed here, BPH 36, 33, and 19, have highly associated with BPH resistance. These genes can be useful for development of resistant variety down the line.
Guru, your slides are not moving.
These, all the works has been already communicated and published in the standard journal. Further, to see the molecular level of resistance, we have conducted a study taking resistant.
Excuse me, Dr. Guru.
Resistant is a Salkati and susceptible germplasm is a TN1. We have seen the proteomic analysis. Here, what are the different kind of protein expressed during the course of infestation from the different time period? 0 as well as up to 6 hours. This study yielded us related to the stress, responsive to stress. Further, to validate the result we get from the sequencing data, we have taken this many number of genes for RT-PCR analysis to validate the study. Among these genes, we have identified. These 9 genes have amplified. Among these 9 genes, 6 genes here I have listed here. These expression only occur during the early infestation, not in the later infestation. Where it's not expressing during the initial hour, but this is another important gene. Here you can see clearly that TN1, this expression not occurred only in the variety Salkati, it's occurred. So this gene can be a potential target. We can go for the controlling of BPH if you are developing some resistant variety. So these host plant resistant works led to development of BPH resistant rice variety, that is CR process of releasing in other states. Other than that, we have identified 2 lines in Naveen background as well as also get released for BPH resistance. Further in the molecular entomology, I have done this complete mitochondrial genome sequenciation. Why I have done this work? Till now, as you know, that in BPH, 4 biotypes are available. Of which biotype 4, still now there is no information available in our gene bank. So this our biotype 4, we have sequenced. It is available in the public database. This will serve as the reference genome. Whoever will be working down the future line, they can use this as a reference genome. And one of the most unique finding we observed here, 2 genetic rearrangement occurred in our population, biotype 4. Here you can see, this is the sample from our study. Here we have observed in the 2 regions, gene rearrangement occurred, as well as one extra copy of the gene present in our sample.
The message that the slides are not moving. Can we please upload again from this slide you can proceed.
Now it's moving.
Online.
Online. Shall I start, sir? Okay. After this, we also observed the nucleotide diversity of this reported gene, 37 mitochondrial genes. Why I am giving this slide here? Among the 37 different genes like we know, COX1 is highly used for identification of DNA barcode and species identification. Other than COX1, like species specificity also is there in this mitochondrial genome. We can use COX2 also for evaluation purpose, but like ATP8 or NAD gene, which can be useful for identification of different biotypes because their nucleotide diversity is very less. Other than BPH, we also sequenced 2 other insects, namely rice gandhi-bug Leptococcus aureteria and green chameleopard Chrysotexis acuta. This also has been communicated. And sir, next slide is not moving. Yeah. Then another important study we done in the BPH, we have identified the different hotspot location in India where BPH severity is more. Those locations we have collected populations. These population used for genetic diversity studies as well as migration pattern study. We have identified the genetic diversity. Here you can see this is the haplotype map analysis. The left one, the pink color shows that South Asia population, and the black one is the Japan population, green one is the China population. We can see here that South Asia population having the close association with the China population, not with the Japan population. The Japan population and our population is entirely different and unique. There may be a reason, till we are not analysts, but what I'm thinking as a researcher, most of our varieties based on Oryza sativa, the Japan variety, maybe it's due to Oryza japonica. So that kind of analysis we have to do in the later period to confirm this result. And the migration pattern also we have analyzed. We identified the bidirectional migration we identified from South Asia to China, from between China to Japan. There is a migration is occurring, but between China and South Asia, we didn't observe any migration pattern through model. But with radar entomology, will be further down the line, we can get this empirical result. And this also has been communicated and published. Another important area is climate change, and I have worked with Dr. Subha Chandra in my PhD in climate change. So that same work I am also continued a little bit further in our institute. We identified the positive effect of climate change means elevated CO2 on BPH. This elevated CO2 increase the multiplication rate of BPH. There are 2 reasons. One is the fecundity. Under elevated CO2, BPH experience more number of fecundity as well as their duration of development is development was more. But in terms of plant phenology, under elevated CO2 condition, the plant CN ratio got changed. That is the one aspect. Another one important aspect is crop canopy circumference, means more amount of growth we have observed under the elevated CO2 condition. Why I am stressing this means, and when we are developing any plant protection methodology in future condition, we have to keep in this character in consideration. For example, now we are using 500 liter of water to spray in the per hectare basis. When the plant canopy is going to increase, so what may be the best spray solution that we have to work on the land. And another one on BPH, here we have identified the different location details. This has been fed to the model to identify what is the future potential distribution of brown band hopper in India. Here you can see at present, BPH having the most suitable area in India is only 7.5% of our total land, but this may increase in the future. For example, in the 2050, if whatever the climate prediction done by IPCC is true, and then it will be going to increase from 15 to 27%. In the year 2070, there is alarming rate. If we are not making any progress to reduce the CHG gas emission, then the infestation may increase up to 50% or more than 50%. And coming to the pesticide, here we have done efficacy study as well as residue study and reduce the point source pollution. And this is the triflumisophorum. This is the unique molecule. Now it is effectively working against BPH. For first time, we have studied the residue dynamics of this molecule, as well as another molecule is colandranate fruit. This is also the highly used molecule in the rice ecosystem. For this molecule, what is the, if you are simulating the condition, natural condition, erratic rainfall is there and different water condition is there. These conditions we have simulated in the laboratory condition. So what will happen to these chemicals? We studied when there is a erratic rainfall, when you are applying this insecticide, if you are not giving minimum 4 hours of period, there is no use in using this insecticide to control any target pest. Another one important study we did to avoid the point source pollution, means we are using the most amount of chemicals, but when we are dumping there, are those chemicals will get leach off and it will go to the river basin as well as the groundwater pollution. So we have developed a model here using the different biomixtures as well as biochar. And we have applied the chemical in the top of the layer. We applied and we observed and we are collecting the leachates every day up to 90 days. Even after 90 days we collected, only 2% of chemical applied in the top reached to the leachate using this biochar as well as our biomixture. That means 98% of the chemical, it get dropped in this biomixture and biochar. So this could be one of the best option. It can be applicable in the field condition. So we can avoid the point source pollution and leaching of this chemical to the runways as well as the river sources. And as efficacy purpose, we have in our previous study, we identified efficacy of this eucalyptus oil against 2 storage pests. We found this is quite effective. So we thought that we developed the nano-emulsion. This nano-emulsion methodology also has been characterized and this has been found effective. And this is the last 2 slides, sir. Under IPM, as you all know, Trichoderma japonicum is one of the best parasites available for controlling OSP. Here we used Ephystia cochneella as well as Citrodoca cereal as the factitious host. We observed, this is an important study you can see here, rather than Carcera sapolonica, when the Trichoderma japonicum reared in Ephystia cochneella, it have increased parasitism rate and the field parasitism against OSP also increased. This is, we identified efficacy of this one important botanicals against BPH. This also has been published. And the last, we also have conducted an extensive survey in Eastern India among around 450 farmers to assess their behavior, why they are using pesticide, different kind of pesticide, what may be the reason. And we found that on their own moral norms, the social pressure is one of the main reason to use. It's not like that I want to use, I observe my field, I want to using. If others also using, so I have also used in my field. So the social pressure also one of the important norms, casing the farmers to spray insecticide. So to break this juncture, knowledge is the one important component that need to be applied in the field condition for judicious application of pesticide. So the summary, I have already discussed all things, so I am not repeating here. And finally, these are the glimpses of some field visit, and these are the products available, commercially available from our institute, from our group. 3 biocontrol agents, and last one is the microbial consortia. It can be used against rice pest and disease, and one app. I am actually participated in this. And finally, I would like to thank all my funding agency for providing the opportunity to conduct this kind of research, and my sincere gratitude to all the mentors, starting from Dr. Ramamurthy sir, Dr. Subhash Chandar, my MSc guide, Dr. Biswajit Paul, my immediate boss, Dr. Mayabini Jana. From there, I learned what I am doing today, as well as my sincere thanks to all my colleagues from Team CPT NRRI, as well as my past students and the present students who are working with me till now. Thank you.
Thank you. Dr. Guru Prasanna, very nice presentation.
Thank you.
You work with genetic diversity, and a lot of diversity is there in your research as well.
Yes, sir.
Right from post-plant resistance to climate change to farmer behavior. So very diverse topics you have been working. And then white-back plant hopper.
Yes,
This is
sir.
in the news recently in Punjab and
Yes, sir.
Northeast, North India,
North India.
especially grass-resistant virus disease. I think your screening of combining markers
Yes,
based.
sir. Actually, I'm mainly focusing on BPH, and our one more scientist is working on WBPH.
WBPH and BPH work will throw some light to some solution for the new emerging problem in rice.
Yes, sir.
Especially in the
Definitely.
North India.
Yes.
Thank you very much.
Thank you, sir. Thank you for the opportunity.
Now I invite Dr. K. Selvaraj to make his presentation as an young entomologist awardee. 12 minutes, please.
Yes, sir. Good afternoon, everybody. Respected dignitaries, eminent entomologists on the dais, up the dais, and as well as online. I am Dr. Selvaraj, a senior scientist working at ICR NBIR. My specialization, whitefly taxonomy, biological control, IPM, and insect ecology and climate change impact assessment. This is my the outcome of the presentation. This is the presentation in the nutshell that my academic profile. Completed my school studies from the government higher secondary school, and my completed BSc agriculture from TNAU, MSc agriculture from CCHAU Haryana, and then I pursued my PhD from IRI, and my initial posting almost 5 years I served at Central Research Institute for Jute and Allied Paper, and then the NBIR from 2016. So after joining my NBIR, I most of my work on in my presentation I covered the whitefly, where I conducted at NBIR, and few studies at my previous institute. Next, please. As we know, the biological university is a great matter of concern for the biodiversity and the Indian economy. The more than 110 invasive insect pests are recorded from India, out of which the whiteflies are major portion of that. In India, more than 469 whiteflies are reported under 71 genera, and out of which the 8 species are invasive whiteflies. Next, please. These are all the invasive whiteflies, 8 invasive whiteflies in the from India, and most of the invasives are recorded from in the coconut ecosystem. Next, almost all the invasive whiteflies are the originated from the this the neotropical origin. And these are all the invasive whiteflies in the distribution in India. Almost since it's the coconut, the most of the invasives are predominant in the coconut ecosystem. So this is very predominant wherever this coconut is cultivated. My first the record is 2016. That is the invasive whitefly, rugose spiraling whitefly, where I most of the work concentrated on that aspect, from the identification to the management, including the technology development. And as the common name indicates, it is like almost look like similar to the earlier the first invasive whitefly, the spiraling whitefly, Allelotechus dispersus, but it's an adult, as well as there are few differences in the taxonomy point of view. And this adult is this is the largest whitefly so far known from in India. And next slide is not moving. Dr. Shashank, so who is there? Yeah. So these are all the major difference, the taxonomic difference between these two the predominant whitefly species in India, where as common name indicate, the scientific name indicate, the rugose appearance in the operculum ventral side. This is the main difference in the particular species. In the adult, they are given the brown band. This is the major difference. And these are all the different life stages of this rugose spiraling whitefly. And then 2019, we have also recorded one more whitefly. That is a form infesting whitefly. It is in dark color, except adult. After from the first instar onwards, it will turn into the dark in color, and it will also secrete a lot of honeydew, and it will make the shooting mold darken shooting mold. It interfering the normal photosynthesis of the form, and this whitefly is also creating major havoc in the South India. And the infestation, even it goes to the up to the 100%. The major well establishment so far, we have the most preference towards the coconut and oil form, and this is a major taxonomic identification as what you call this Aliotrichus attratus, and which is different from this closely associated or the closest species Aliotrichus tracheides species, which is Solana whitefly, which is also predominant on mainly on the Solanaceous crops. These are all the different life stages of this particular form infesting whitefly. And next, full slide is not visible. This is the woolly whitefly. This is we have recorded in November 2019, and this is although this in the native countries is a series based on citrus, but in India so far we have recorded only on guava, and is well distributed in almost in the entire South India. And same as the symptom of damage. We can see that how the colonies, the diagnostic characters, and here also infestation even reached to up to the 100% complete coverage of the host plant, and even complete defoliation occurred. And this is the different life stages of this particular whitefly, and this is the taxonomic character of this particular whitefly, where the margin is having the coarse teeth as compared to this Solana whitefly. And few more there are whitefly, maybe for the those who are there are all new species, so I have included some of the life stages of this Solana whitefly, where this is very predominant in the Solanaceous and Convolvulaceae plants. It's very serious issues, and many of these many of the state I will come in the distribution. And this is this bandar nesting whitefly, where the make nest, and they like bird like nest using the wax material, and this is the bandar nesting. This is nesting whitefly. That is Paralidotus mini, and there are a lot some difference in the adult, where the X-shape marking in the adult, and which is absent in the mini. And this is another legume feeding whitefly. This is also emerging in India, and this is the Tetralidotus acaciae. Is mostly host preference towards the legume, most of the legume crops. And all this work we have published consolidated in the Phytoparasitica recently, and also the many articles in the newspaper. And we have also studied this distribution map, the current and future distribution using Maxent model. And these studies so far we have carried out only for the rugose, and other species we are in the process of developing the model. And this paper is published in Journal of Agrometeorology. And as the whiteflies, as you know, that most of the whiteflies are adults are lethargic in nature, but it's mostly mediated through the human being, the movement of infested seedling material from the infested seedling from one portion to another portion. Almost all the whiteflies are highly polypagous, and where we can see that how the host range of all the invasive whitefly, they are the prepared host plant. And there are some of the host plant, the rugose infestation on different host plant. We can see that in globally more than 120 host plant is recorded. In India, more than 45 host plant I have recorded. And this is a coexistence with another very uncommon phenomena, where as in ecology, as the more than one species may not be coexist in the same niche, but it's not in case of this whitefly. Even in the native countries also the same behavior, as these are coexisting each other, even in the same plant, in the same leaf, even in the same colony, they are coexist. We can see in the images, almost all the crop, there may be some novel mechanism which is managing this the coexistence, the kind of the survival mechanism and exploiting the ecosystem. And this is the impact, where you can see how the normal photosynthesis of the plant, and the depleting the nutrition and water from the plant. And this is seriously affecting even this Aliotrichus tracheides act as a vector. and it's excrete a lot of honeydew. You can see the complete shooting wall development, and the even complete drying and drooping of infestation. The infested leaf will be observed for this whitefly. And come to the natural enemy, most of these whiteflies are reached in the outbreak situation. As we can see that we have recorded only parasitic for dispersers and atreus and rugose, but other whitefly we don't have any potential parasitic in India. So it shows that they are maybe entered in India without any their natural enemy complex. And these are all the natural enemies where rugose spiraling whitefly. That is this is the very potential parasitide Encarse godolope up to the 82% parasitism. Rugose and spiraling whitefly, Encarse dispersa and rugose and spiraling whitefly. And this parasite recently we recorded Encarsia cubensis on palm infesting whitefly, and this paper is communicated in Phytoparasitica. It's under review. And except there are many like indigenous predators. And we have also identified one potential entomopathogenic fungus, Eisseria pemmicerosea, which kill almost all the life stages of all the invasive whiteflies and giving up to the 80 to 78% under field condition. This we have developed further different formulation, and we have also commercialized this Eisseria pemmicerosea and to 4 companies. And these are all the different natural enemies in the associated with these invasive whiteflies. And sorry. This is the, as I said, this is the new parasitide we have recovered from the saleratrical traders. Although we have last 2 years, we are attempting to release the 2 parasitide from Hawaii. Meantime, we have recovered this parasitide, recovered and identified. It's also giving very good parasitism, up to the 68% so far, and probably I expect that it may be in the reaching more than 80% in future, because once the prevailing condition, once it established in India. And these are 2 technologies we have developed for this rugose spiraling whitefly. For Encarse godolope, redistribution is nothing but the field insect technique. We have to conserve in the in situ conservation and distribute to the adjoining or the next wherever the infestation is occurring. The parasitized nymphs will be redistribution, because the pest move during the adult stage, so they will detach from the host insect. So we need to favor for the redistribution of this parasitide, the inoculative release. Then in inundative release, we have also standardized the mass production technique for this particular parasitide, and we are recommending the more than standardized for the 600 parasitized nymphs, minimum 2 spray to bring down the pest population. And we have also standardized the release technique for this particular parasitide. And we have also the more emphasis we also given for conservation biological control. As we know, initially during our madam time, we have conducted one brainstorming session, and we have called all the stakeholders and first to declare the pesticide holiday. This is the most important achievement. Based on that, so far none of our either the state government or the central institution, those who are working in the whiteflies, we are not recommending any of the insecticides. Still now this formulas we are following. And we have also formula in the NBR, we have standardized the habitat manipulation technique using the Canna indica plant or banana as the intercrop, which is also giving the very good control for this parasitide. And then we have huge extension work we have conducted using the 60 extension program and different trainings we have carried out. And this formulation Eisseria pemmicerosea we have standardized, and we have formulated different formulations, and we have also evaluated against other non-target organism or different Pampex mori, Pseudomallada, the goniosis, and this is the different the infection of this particular Eisseria. And this we have supplied this both predator as well as the parasitide and the Eisseria we have distributed to different stakeholders in India for their augmentation and spraying. And then we have conducted several trainings for the different on-farm production to department officials, farmers. And we have established this production unit in Andhra Pradesh and one at Tamil Nadu. And one mobile app we have developed for the Satpada RSW. And this we have some studies on genetic groups of Bemisia dapasi, and 2 genetic group we have determined, and the ACA1 and ACA21, and more than 80 DNA sequences submitted for the whitefly and their natural enemies. Okay, and this is covered documentation at my previous institution, and there are some studies at my previous institution. The climate change studies, thermal constant, and some past paper influence on the 2 important pests, Spilosoma and mite. And this all one germ blossom also associated with one germ blossom registration. This is completely resistance against Spilosoma. And this is one JAP expert. This is the expert system for the jute and allied papers. And this is all my technologies and their trainings and several demonstration activities. We can see that most of my times will spend for the teaching the farmers and other stakeholders. and the one deputed to Shark training for attending the IPM training, and also I deputed to Australia for Brisbane, Australia to get the hands-on training on parasitide, whitefly parasitide, and also they guided one teaching, associated one teaching in Ramakrishna Mission and guided two students, and also the master trainer.
Thank you.
Thank you, sir. Thank you.
Thank you very much. Nice presentation.
Thank you.
Dr. Selvaraj, I think you have done very good work on the invasive Rugose whitefly and other whitefly
Yes, sir.
species, and also the distribution maps, and also commercialized
Yes,
the
sir. Both the technology
solutions for
commercialized.
this whitefly, and also habitat management and pesticide holiday. I think you and madam have really held the fort, because the tendency is to go for insecticide applications. That would have actually endangered much more than done much more harm than good. Thank you very much for your nice work. Thank you. We don't have time for questions.
Okay,
Thank you. Now I invite Dr. Shashank to make his presentation. 12 minutes, please.
A very good afternoon to everyone, all the dignitaries and all the senior entomologists in the auditorium, online and offline. So today I'm going to talk about my journey into the insect world. Actually, from my master's onwards, it's been a like a roller coaster journey, being an agricultural entomologist and taking a taxonomy and staying true to your profession and also for passion. So I'm presenting my work in three different subheadings. The first one is what I exactly started with, taxonomic revisions, and which I am covering. And after that, the service we are providing, insect diagnostics and monitoring and management of insect invasive pests. The first foundation for my taxonomic work started at Bapatla, and my boss, I'm the first student for my boss, MSV Chelam. He's a leafhopper specialist, and my work was to study leafhopper and plant hopper from coastal Karnataka. And also because at that time, most of the agricultural universities wanted us to work in some management aspects. So the last word has been included in my topic, which is completely different from my taxonomy work. However, I could work on leafhopper and plant hopper taxonomy, and I could study, because MSc is just a training. I could study around more than 20 leafhopper species and about 6 plant hopper species, which are important to rice ecosystem. At the same time, this is my first paper, which, because I'm really excited to tell, at that time, being an MSc student, publishing one single paper was really happy moment. So my first paper was on evaluation of novel insecticides on plant hopper and leafhoppers. After that, I have joined for PhD to GKVK, US Bangalore, and my topic was on understanding the cryptic species, actually host associated cryptic species of Conogathus punctiferalis. So to address this issue, we selected an integrated approach, and I'm really fortunate enough to work with all the stalwarts at that time and still now. So for every aspect, we have selected a person who is really good at that. So we have addressed all the issues a common entomologist will ask to differentiate two species. The first thing, in Conogathus punctiferalis, which is feeding on castor and cardamom, we have studied morphologically, and we found that there are some differences. And at the same time, we also gone into the DNA barcoding, and we found that there is a difference between the species, between the host associated populations. And later, we have also studied behavioral aspects of it, taking pheromone as a basis. Before doing for pheromonal experiments, we tried to understand when the pheromone is produced. So when the moth will emerge. So we all studied daily adult emergence rhythms and how many days the adult female and male moths will survive. all this data, and we published in very good journals. And also we have done interbreeding studies. So there are a lot of studies we done, and we concluded we have done interbreeding studies. After that, we have also evaluated pheromone molecules, both abdominal gland extracts and also available synthetic extracts. And we found that both the populations, they are different at minor molecule level rather than the major molecule level. And these are really interesting study, but however, I could not publish as the cardamom feeding population as a new species because of accessibility to study the type specimen. So in 2018, we could publish this Conogathus iadrensis, which is a species now recognized species, which only feed on cardamom. And in this paper, because I was in IRA, I could add a lot of data to this paper, and we have done whole genus revision of Conogathus with a lot of molecular data. And it has been published in 2018. After that, in 2012, I have joined IRA as a scientist. I'm really fortunate, and thanks to Dr. Ramurthy sir, who brought me to IRA, and I joined NPC. After that, I started working on Lepidoptera taxonomy. So my first project is DST fast track grant, which I got for taxonomy of Flucinae. And this is one of the important pest group, and we could study all the species which occur in agriculturally important ecosystems. And we have revised this complete subfamily, and we have published this monograph in Zootaxa. With first time we have generated all the reference DNA barcodes. That means each specimen have that barcode along with the taxonomy records. And you can see these are just plates, which a lot of data has been put into this. And my student Twinkle, she worked on this. And we have also published a website so that all people can just go to that and check for a morphological identification. Another work which we recently published is moths of Delhi. When I came to Delhi at 2012, I started studying the moths of Delhi, and we have published a huge data set of more than 2,000 specimens, along with like illustrations of 195 species, and it's a data of 8 years. Recently, one of my MSc student, Komal, she helped me in publishing this. And of course, because it's Delhi, you get a lot of media attention. And the another work I have started is understanding because I have a background with pheromones, so I always look taxonomy in a different way. So we had a doubt that the sensilla is very important to understand the pheromone chemistry, but we don't have a lot of data on morphology of sensilla. So we took this work, and along with Rani, she's at IHR, and we have studied the morphological characterization of Aeris vetella, and we published this in 2021, and we found that 9 distinct types of sensillae occur in Indian population of Aeris vetella, and that's very unique. And this paper, this paper has been published in Micron, which is mostly a microscopic journal. But the good thing is this paper has been most popular paper till now after the publication. I'm really happy for that, because it's very including publication. And also the expertise in molecular entomology paid me many times, and I collaborated with my collaborator, my colleague, Dr. Naresh Mishram, and we started understanding 3 important aspects. One is, anyway, we are describing a lot of, as a taxonomist, we are describing a lot of new species. But is that all the new species just we are describing? Like, we wanted to understand the relationship, and we had to go ahead with a lot of new novel data. So we started collaborating. I started collaborating with Leafhopper Group, and we have published series of publications with Dr. Naresh Mishram. You can see on all these, because we have added a lot of molecular data, we got this published in very good journals. And we also got around 4 research external funded projects for this work. Until now, I have, like, in different publication, I have, of course, have published small new records, new species and all. And the next responsibility which we have at IRA is insect diagnostics. And of course, every year we get a lot of samples for insect diagnostics. And I have like on an average, I have contributed around 200 specimens per year and more than 40 institutes. This is a good way to collaborate with people. People send specimens to you and you interact with them. And this is, I really enjoy this. And the next is research expedition. Being a taxonomist, everybody want to go to field. Because I got external funded projects, I could explore so many places. And along with my research, I'm doing expeditions also, and also insect collection management, because National Pusa collection is very old and we have to manage the already existing collections. And now we have also published, we have this National Pusa collection website also. Now we are slowly digitizing all our collections and we want to go online. In 2014, I've started working on invasive insects. It's a coincidence, being a insect diagnostic center, I got this specimen from Dr. Chandrasekhar, Pune. And so slowly I started working on Tuta absoluta. And this is our first record, which is published in 2015 in Indian Journal of Entomology. And later we have also started monitoring of this pest throughout the India, along with my collaborators. And we started working on different aspects of Tuta absoluta. Because it's a new pest, every aspect is important. So we published our first molecular genetics study in Tribotech. And later, along with Dr. Fond, I also because when you survey a lot of area, you have this presence and absence data. So we could generate a lot of presence and absence data. Dr. Fond is good at modeling. So I just collaborated with Dr. Fond and we could publish and we could understand the ecological mesh modeling and how this pest can be dangerous. And also we first, when it entered the India, we first published a rapid action plan in ICR News so that people can adopt the rapid action plan, and also for farmers education. Anyway, fortunately in North India, it has not become so severe compared to South India. But at that time, we were ready with whatever things we wanted to do. And because of this work, I also got a lot of global collaborations, and we published 2 important review papers for Tuta absoluta, which has been cited highly. And one of the paper which published recently, I'm the 47th author. It's a global analysis of IPM package we have done for 37 countries. It's a wonderful paper. When we work with those people, we will also understand in what way we have to look into these problems. And also the Nepal Agriculture Research Council scientists also approached us for identification and what we have done on Tuta. So we could collaborate with them, we could share data, and we published small papers with them. And yes, sir, I'm done. And this is my only Tuta which was a Lepidopteran and also which we could identify. And we covered all the aspects of Tuta absoluta except management, because I'm not into that. So I thank all the collaborators who helped me in this. And also, being a biodiversity worker, a taxonomy worker, I also get involved in a lot of outreach programs and also in citizen science programs. And I thank all my faculty and all my research scholars, because most of the work at IRI, which will be helped by my students. And I had a really good team. Every time I'm so lucky that I get very good students. And I got a good external funding support from the beginning. And being a taxonomist, getting recognition is always good because it encourages you to do more. And recently I got different awards, of course. But I want to mention this Agriculture Today Award, which exclusively given to an agriculture scientist, because our work in invasive insects and especially insect diagnosis, they have recognized. I was really happy that somebody is really recognizing insect diagnosis. So I thank all my collaborators and funding agencies. And there are so many people who helped me to come here and present in front of you. I thank everyone. Thank you.
Thank you, Dr. Shashank. I think
Thank you, sir.
the combined use science to communicate your results and then connect with the public. And whatever you have done as a taxonomist, you have been taking it to the end users
Thank you, sir.
in different ways and means, and done lot of collaborative network research, tapping the potential of each other and then coming out with, and that eventually led to global connections as well.
Yes.
Thank you, and
Thank you,
commend
sir.
you on your work,
Thank
and
you,
all
sir.
the best wishes to you.
Thank you very much.
Thank you.
Thanks.
So we would like to go with the last presentation. Best PhD thesis awardee, Dr. B. Karianna.
Namaste to all. I'm very happy to be here and to present my PhD achievements. Actually, for my masters, I started with taxonomy. I worked on Cerambicidae, and I had one monograph on longon beetle of India. And later, I thought to go for molecular aspect, and I just approached my professor, my earlier guide, co-guide, Dr. Mohan sir, who is in NBR. So then he agreed, I'm a student of US Raichur. So where I enrolled for PhD under under supervision of Dr. A. Prabhuras sir. So I just approached Mohan sir, just I want to work on this, and could you please guide me regarding? And we had one, sir is having one project related to genomic resources, and it's huge project, and there they have already had one draft genome of lucinodes. They were already developed that, and they had that info, and he told we can work on it. Only thing is you have to learn. So then okay, I took that projects, and we just collaborated with that particular project. And initially sir was given it was a simple projects, simple thesis assessment, only to study the identification and functional analysis of cytochrome P50 and their resistant effect on in Lucinata subanalysis against different insecticide like that. But in the course of time, because of the resource availability and facilities provided by ICR NBR, made me to take a few more group of genes glutathione esteransferases as well as carboxyl esterases. By keeping that in mind, I went with a deeper expedition, deeper research, identification and functional analysis of genes which are involved in insecticide resistance in Lucinata subanalysis. That was my complete and comprehensive research title which I took. And the base for this particular thesis to take up was we know that Lucinata subanalysis is one of the important pests, and it is causing so much havoc and enormous damage in brinjal, and regularly farmers are using so much of insecticide on it, and it's causing nearly up to 60 to 90% damage. So by keeping that in mind, I took this as a project. And so it was earlier going exactly to the molecular aspect will not be so convince you, let me take some even ecological part as well as some toxicological part that will give correlative aspects, so that we can easily conclude the topic like that. So then I went with different what's called objectives. First objective was population dynamic of Lucinata subanalysis against different cultivars, and followed by insecticide it's called resistant population identification across the India. And by using that different screening of the different genes, for example cytochrome P50, glutathione esteransferases and carboxyl esterases, and followed by their expression study, because I have collected around 5 field population across the India, and they're resistant over the isophemal line, which are already maintaining in ICR NBR since 5 to 6 years. So that means I have done, and followed by knockdown of the key resistant genes, whatever I got from the expression study. So that I went for further analysis, and I got so conclusive data, and I will express one by one. The first objective was population dynamic of Lucinata subanalysis against different cultivars. So here I have took some 3 types of cultivars, one 2 hybrids and 1 improved variety, that is Manjari Gota, and followed by 4 local varieties. So when I took the 2-year consecutive data, and I got the result that the resistant local variety, that is Iranagere, was showed very less infestation by this Lucinodes sabanalis compared to other varieties. That is about 23%. And the some of the weather parameters like maximum temperature, wind, and morning relative humidity and minimum temperature shown negative effect on the infestation. And followed by like population distribution, when I studied, like there was a bimodal distribution of the population was observed. Like so at the verge of Rabi season, the population was decreased, but at the end of Kharif, the population was increased. So it was an indication that like the summer the summer population will act as an inoculum for the their infestation, like that. So this so here is a pictorial representation what we can see here. That is a bimodal representation. And followed by that, next objective was like identification of the resident population across the India. So the population like which I collected was Bhubaneswar, Dharmapuri, Pune, Raichur, and Varanasi. So this was studied again as the different group of insecticide like from OP as well as carbamate, neonicotinoid, and like a new molecule like flubendiamide and chlorantraniliprole. So the study was showed like the population was showing so much of resistance towards the frequently used insecticide. For example, here the frequently used one such insecticide was like phenolhalate and even flubendiamide. The ratio was like 29.5 to 303 fold resistant against the flubendiamide. So this was like in an initial instinct that we can there will be chances for further study related to the expression. So then the next objective what I was took like identification of their enzyme production within the population, because I have collected the population from the different locations. So how the production of the enzymes, so that enzyme whether really involved in detoxification of the insecticide. So when I went for the studying that enzymes interactions with detoxifications, so I have identified like or demethylase of cytochrome P40 monoxygenase. It was like high overproduced in the population which I collected from the field, then compared to the isofemale lines which we are maintaining in the ICR NBR. And same was even observed for the carboxyl esterases as well as glutathione esterases. So here is the like the different fold changes was observed, like higher concentration of the these enzymes in the field population then compared to the like isofemales, like. And even carboxyl esterases, it's quite tough to identify based on their production production in the system. So I went with like vertical gel analysis, where I found like 2 more extra low molecular high molecular weight bands, so which indicate that in field population the carboxyl esterases was more than compared to the like isofemale line what we had isofemale line. And the slide is not moving. So actually this was given an idea that like there are genes like enzyme control governing genes which are involved in this which may be involved in insecticide resistance. So by keeping all this basic informations, so and even the data of Lucinodes sabanalis, so I went for like identification of the genes. Particularly that next aspect was like identification of the different genes which are present in the Lucinodes sabanalis from the whole genome draft. So here before entering into the actual genes identification, so there is a need of standardization. For the standardization, like there is a need of like housekeeping genes. So I have selected around 9 housekeeping genes from the different functional aspects. So in that, like 28 SR3 as well as GAPDs are 2 important genes. I will explain in the next objectives. And regarding like identification of the cytochrome P50 and glutathione esterases as well as carboxyl esterases. So there was like, because of the huge sequences, like we have to identify. So we found so we followed the series of procedures, and we found that like for cytochrome P50 around 74 genes, 42 genes were identified from the Lucinodes sabanalis, which are grouped into 4 clans, which are very important. Among the 4 clans, like the clans like the genes which belongs to clan 3 and clan 4 are like importantly involved in insecticide resistance. And here, like the genes like these 4 genes were highly expressed in Lucinodes sabanalis field collected population. That I will explain in the future object, and followed by glutathione S transferases. The same method was followed, and here also there were we identified the 94 genes of carboxyl esterases from the Lucinda savannalis, which we grouped under different subgroups, and followed by glutathione S transferases. Here the group was the putative genes were grouped into 6, and so in this 34 genes, we have means I particularly identified 24 were having insecticide resistant property. During this study only, I have identified which are all the genes which are involved in insecticide resistant based on the BLASTX NCBI database, where the 90% query coverage and homology. So by using that, I identified particularly the finding says that there were 74 cytochrome P50 genes present in Lucinda savannalis, and 25 in 34 glutathione S transferases, as well as 94 carboxyl esterases. Among them, 34 resistant from the CYP genes, and 16 from the carboxyl esterases, and 24 from glutathione S transferases. So further, that next study was expression study. So whatever the genes we have studied, and even based on the database as well as homology. So we have I have found that there are some genes which are having insecticide resistant nature, or detoxification nature. So further I went for the expression study of them. So so here first I went I colored the genes, and I prepared the primer for them by using primer 3+ software. So here around for particularly cytochrome P50. So sorry, before we're going for that cytochrome P50, normalization was done for housekeeping genes. Particularly here, 2 genes I told 28 SR3 as well as JAPDH. They they were stably expressed in all the stages as well as different conditions. So those things we took as a housekeeping genes for the standardization, the further expression study, and so these are the 2 important genes, and the further expression study for cytochrome P50, I told you already that. So the 34 genes was identified, and their expression study was conducted. Here in from all the 5 field collected population across the India, the 4 genes were stably expressed in all the resistant population over the isophemal line which we maintained in ICR NBR. So those genes even that heat map also was studied, and these genes were typically commonly expressed in all the populations, and particularly these 2 genes CYP324 F1 as well as CYP306 A1 are quite important, because this CYP306 A1 one such kind of a genes which I identify first time from the animal kingdom. And for particularly the finding says that these genes are having highest production, highest transcript in the resistant population, field collected population, than compared to the isophemal line what we have maintained in ICR NBR. The followed by the same way, even for glutathione S transferases which I conducted in the 24 genome, 24 genes I have selected for the preparation of what's called primers, and further their expression study was conducted. And the expression study implies that the expression study implies that there were there were 2 genes which are stably expressed in case of glutathione S transferases, and further it's particularly glutathione transferases, this was 6 genes, because the glutathione S transferases that problem is duplication problem will be there. The expression study and further analysis will be very tough. Hence this 6 genes whatever we have studied, and which I just documented it, and further I went for carboxyl esterases. In this carboxyl esterases, total 94 genes were there, and among those 94 genes, only 16 genes were matching with the insecticide resistant from the different populations. So those 16 genes, why? Because among the 94 genes, more major genes are involved in behavioral aspect rather than this metabolic involvement, metabolic functions. So here the study the result implies that only 2 genes which they shown stable expression across all the field population, but they were not so prominent compared to the cytochrome P50 and glutathione S transferases. So those genes were contigged 2 contigs, and followed by that next method was what I followed was knockdown of the genes. particularly here, that final aspect, what I took was two genes. I told you, like, 306A1 and 324F1. These genes were I selected for the further knockdown study. So where I prepared the dsRNA of these, and I used the further method, like, feeding, flower bud feeding method. So here the flower bud feeding method, when I injected to the population which is showed more resistant from the field, like, for example, Dharmapuri population showed highest resistant in the field condition based on my previous study. So that population I used, and one susceptible population, that Bangalore population I took, NBR population, as well as now one positive control and negative control I studied. And the study implies, like, I have provided different concentration of the dsRNA from 0.5, 0.05, 0.05, 0.25 and 0.05, like that. And the study implies that there was like highest down regulation was seen in case of highest concentration. For example, here the 1 microliter concentration, the gene expression was very less. The same thing was even studied for the other gene also, for the 306A1 also. And even when I studied, like, mortality, so there was, like, 20 to 30% mortality was observed, because it is not so prominent, but there was a mortality because of these particular genes, because these genes are involved in detoxification.
Please summarize here.
Yes, sir. Yes, sir. That's all, sir. Completed. Yeah. So it is in the final thing was like the study implies that there are genes which are involved in insecticide resistance. Like, there are group of enzymes and which are governed by genes. So the important aspect is, like, they will govern the insect activities and detoxify the insecticide which we apply regularly. So the best outcome from is, like, some of the genes which already there, and that was already validated by using the dsRNA, and which can be used for the future development of the novel molecules. And the study come up with some good outcomes. For example, the publications from the frontiers in physiology and biotechnology and applied biochemistry and biology. So these are, like, some important publications which came through my PhD thesis. And I sincere thanks to two institute which continuously supported me during my PhD course, that is US Raichur as well as ICR NBR. And I thank my wholeheartedly my supervisor, Dr. A. Prabhurai sir, and Dr. M. Mohan sir. And thank you, ESI, for considering me for this award. Thank you.
Nice presentation, Dr. Kariyana. Very important topic.
Yes, sir.
And you collected this population from Dharmapuri.
Yes, sir.
Where highly intensive cultivation of vegetables, especially brinjal, takes place.
Yes, sir.
Almost precision vegetable cultivation is practiced there, and input use is very high.
Yes.
Ultimately, your your solution that you are going to provide, dsRNA or whatever technology, will be useful for them.
Yes,
Thank
sir.
you very much.
Thank you so much. Thank you.
Now we are through with the presentations. She will make the.
Congratulations to all the ESI awardees. Now I would like to invite Dr. YG Prasad to present the award for today's awardees. I invite Dr. Selvarajan from NBIR to receive the awards on behalf of Dr. Ankita Gupta, Madam.